going with you, so I guess it's a good question.
All right.
It's a privilege after so many years of having to have all these seminars via Zoom, which is still a very useful resource, but it's nice to have speaking in person, and it's really nice to have no other than Jasmine Foo, who I was joking before that didn't need an introduction, but just for the sake of appearances, I'll give you an introduction. Jasmine is a mathematician. She actually has a bachelor's, a master's, and a PhD from Brown University in mathematics. She did her postdoc under Francisca Maker, first in Sloan Kettering, and then at Dana-Farber. And from there on, she moved to Minnesota, where she's now a full professor at the University of Minnesota. She has also an affiliation with the Institute of Math and Applications, where she used to be the deputy director at the UMN, the math department. She also is the associate head. She has a lot of accolades. Other than being a full professor, she was a Fulbright awardee that allowed her to go and visit the University of Oslo. She has an NSF Career Award, many more accolades that I don't think I have a time to describe right now, but these are some of the... Your voice in the background?
Raphael,
yeah.
Mute yourself.
Yeah.
Sorry.
Don't be sorry, yes, I'm muting myself. Okay.
All right. I'm switched. I'm trying to figure out how to switch my display. This is what we want. We don't want that.
No.
No, it used to be the display thing, but that's very up there. So,
Sunny, we need a bigger collaboratorium. David,
Yeah?
it could also be on the Windows computer.
Oh, I'm pretty sure it is.
That's me. Well, just to finish that, Jasmine has over 50 publications and has been granted awards and grants from the NIH, the NSF, and the state of Norway. So, yeah, with further ado, please, Jasmine.
thank you. Bear with me a minute while I figure out how to
yes.
get the right screen.
So you've been sharing just the presentation file, but you share desktop, like, I feel like here.
Oh, okay.
Yeah, you just click over there. Yes.
And I'll just, yeah, that's good. That thing there.
What do you see?
Yes,
you did.
That's okay.
Yeah.
All right.
Yeah.
Oh, okay. That's from that machine. Never mind.
Yes.
Got it. All right. Well, thank you very much for the very nice introduction. And I'm really excited to be here. This is my first time at Moffitt, and one of the very few in-person presentations I've given since that which shall not be named happened. And so I'll be talking to you today about some computational methods we've been working on in our group for inferring some tumor evolution parameters and some things about tumor heterogeneity from some data. And so this is just a quick outline of the talk. So the first part will involve looking at drug response heterogeneity, so kind of a phenotypic as opposed to genomic heterogeneity perspective from patient samples. And then in the second part, I will be talking about looking at the state frequency spectrum, which is genomic data, of course, and trying to infer some parameters of tumor evolution from the small frequency end. Okay, so in part one, and I'm stealing this slide from my collaborator, Alvaro, who made this awesome slide. A lot of people have worked on the work that I'm going to show you, and from Norway. This started while we were on sabbatical in Norway, and I just want to point out Evan, who is a current graduate student and has done a lot of the things that you'll see in the talk. But there really has been an awesome collaboration with a bunch of people. Okay, so I want to start out by just kind of talking a little bit about high-throughput drug screening. I'm sure most of you are familiar with this. So you have patient tumor samples that are collected and then run in a platform where they can be screened for response to a collection of different therapies. And the idea is that, you know, you kind of look and see how these cells respond to different drugs and use that to make some decisions about that patient's treatment. So one of the outputs, or one of the main types of outputs that you get out of this kind of screening is drug response curves. So here you're looking at relative cell survival as a function of drug concentration at different points in time, so different durations of the experiment.
So what kind of tumor samples are these, and how do you know that the cells are tumor cells and not like fibroblasts? So if you chop up most of the tumors, you'll get fibroblasts growing out. Tumor cells don't quite grow.
Yeah, so like sort of, you know, in this slide, I'm sort of talking generically about drug screens. I'll talk a little bit more about some of the tumor samples that we used, which were from multiple myeloma later on. But I have to confess that I myself know very little about the processes that you described, but our collaborators, I know, did a lot of work to kind of think about those kinds of questions. For these viability curves, the viability has a couple of different formulations that you can find in the literature, and this is one. So, for example, you might be looking at cell count under some dose B at particular time t. So t is the duration of the experiment. So that's the numerator, and then this thing is normalized by the cell count at time t if you had no drug. So that's a pretty standard formulation of cell viability, and so what you might be plotting in one of these dose response curves might be viability over time, for example. Okay, so I want to talk a little bit in detail about these viability curves. So in practice, people often will fit them to a hill function. So those are these sort of smoother curves that you see here, and the markers are the data. And the hill function, which I've shown here, has a couple of parameters. So B kind of controls the maximal effect of the drug, so in some sense the lower asymptote, and then E kind of controls where this thing is going to dip down, and then N controls the steepness of this dip. Right, so with these several parameters, you can approximately fit a lot of different types of data that you might see. And a lot of times, or most of the time, people kind of are working more with some summary metrics of this fitted viability curve. So, for example, the IC50 or the EC50, which sort of loosely speaking, you can think of as a concentration at which 50% of the viability is reached, or 50% of the maximal effect, or something like that. Another common summary metric, is the AUC, which is the area under the viability curve in the region that you're testing, so within the range of doses that you're testing, for example. So the area under the curve in this green region here. So one of the things that I think is a little bit of a drawback to some of these metrics is that the viability curve is typically sort of empirically known to be dependent on the duration of the experiment. So here you can see as time goes on, this thing is sort of moving down, in some cases moving to the left. And so that's obviously going to affect these summary metrics as well, and of course these things are going to be affected by experimental conditions, things like that. So we got interested in some alternative metrics for drug sensitivity that were more based on growth rate as opposed to viability. In particular, in a paper in 2016, Hafner and colleagues, they proposed GR, which is the ratio of the growth rate under treated and controlled conditions, normalized to one cell division, and then a summary metric of that curve, which is the GR50, the concentration at which half the maximal effect of the growth rate inhibition is achieved. And
thanks for help.
Okay. Let's see. I have to. Let's see. I don't.
It's a question. Why does cell survival increase? No, it's just that.
Oh, that's on yours. Okay.
Yes.
Okay. All right. I'll just finish my slide and then I'll read that question. So these alternative growth rate metrics have the property that they're not going to be dependent on the length of the duration of the experiment and some of these other conditions. And so in this talk, we'll be considering the GR50 as the drug sensitivity metric of choice. Okay, how do we get that question back up? Let's see.
So he's asking why it increases, the cell survival increases at low concentration.
Oh, in this data, this particular data set, I am not entirely familiar with, I just sort of pulled this as an example of cell viability. So this isn't data that we produce. I can't exactly answer that. My guess is that it's something to do with the normalization, because all the data points, all the data that converge at 100.
But you mentioned that, like, in this essay, you start with 100% is the number of cells that you started before the treatment. So in this case, like any growth would.
Right. Yeah.
They can grow
So it's probably like some delay in the amount of time it takes for the drug.
But then this is very low concentration, so maybe they don't really care and they can grow.
Right.
But it's also not uncommon to have the cells grow better in low concentrations of the drug than without of the drug if they are resistant, because basically it moves them to optimal signaling.
Right. Makes sense. Yeah. All right, here we go. Okay, so coming back to drug screening, so the kind of picture becomes a little bit more complicated when you think about the heterogeneity of patient tumor samples that you might be putting into the drug screening. And so now maybe you get out of this a viability curve that is, or a growth rate curve that is a little bit less easy to interpret, because really it's representing the viability or the drug response of a mixed population, and may not actually, there may not be a single cell in your population that has this actual drug response curve. Yeah.
It's probably an incredibly simple answer, but I'm just currently can't figure out how would one get a growth rate out of these experiments. You replace the IC50 with the growth rate as a function of the concentration. How do you get the growth rate?
So you basically can take so if you think about, how we get the viability of all those measurements of cell count over time. So you can use those time series to calculate growth rate.
So there is a full time series behind it.
Yeah. And there would be any way to calculate the.
And then there is the assumption that the growth rate is constant and just affected by the drug concentration. Right.
Okay. So the question we got interested in is, if we have viability data from this sort of heterogeneous patient sample, is there a way to deconvolve this data and determine, whether or not there are multiple subpopulations? If so, how many, and what are their individual drug sensitivities? So this is all sort of predicated on, each particular drug you want to do this deconvolution. Okay, so to approach this problem, we used a very simple exponential growth model to represent the growth of a single population of cells with a homogeneous drug response. So we have a x naught and then exponential term, and this slightly more complicated term is the growth rate, alpha plus log of the Hill function, essentially. I'll get to that in a second why we chose that, but for now, let's take that as the model for a single population, and then we want to build that up to having multiple populations. So now we have x sub I being the population size for the ICE population of cells under drug D at time t, and now we have a vector representing all I populations, and this vector kind of starts out at time 0 with, say, chi I being the initial fraction of population I, and then we evolve this essentially by considering the exponential growth, but also potentially interactions between populations of cells. So I'm not going to go too much into those in this talk, and all the results that I will be showing you assume no interactions, but we did also consider in the sort of general framework population interactions. Okay, so coming back to this, why do we choose this alpha plus log H for the growth rate structure? Mainly because we wanted to retain or we wanted to acknowledge the fact that, for many decades, people have used the Hill function to represent to fit empirical drug response curves. So we wanted to make sure that for a homogeneous population, we're going to capture that Hill function viability shape, but also it shows when you calculate the viability using this model, it has the same dependence on time that we expect. So here, time increases as we move from blue to green, and we're plotting the viability that we would get out of this model. And we see the same behavior, that the viability curves will move down and a bit to the left. So it yields the experimentally observed viability behavior. And importantly, just putting down that growth rate structure turns out to be what enables us to do that deconvolution problem, because we're now sort of constraining the dynamics of the population to sufficiently to be able to make those inferences.
You don't explicitly model depth.
No, this is net growth, essentially. So we're just modeling how many live cells there are. Okay, so in terms of the statistical framework, coming back to the data, we're going to be looking at, from these drug screens, an observed cell number at a particular dose for a particular replicate for a particular time point. So that's the big matrix of data points that we have. And our underlying statistical model is basically this population dynamic model that I just told you about, plus some noise term, right? And so, and then from there, this follows a basic maximum likelihood approach where we say, okay, under a particular set of parameters, so the parameters here, just to remind you, are the dose response parameters for the Hill function for each subpopulation, the initial frequency of each subpopulation. So those are the parameters. So for a particular set of parameters, what is the likelihood of seeing the data that we observed, and then optimizing over the parameter set. And then doing that over and over again for each number of subpopulations. So for homogeneous population, for two populations, and so on. And then we perform a model selection to sort of pick out, how many populations do we think there are, and then we have all of these optimized parameters that'll tell us both the frequency of each subpopulation and the drug response. And I just have a little note here on the type of data, type of observations that we can get in terms of the cell number, and just point out the fact that there's a variety of different experimental techniques. We worked with a number of them, and it's really important to consider what experimental techniques, because well, for a number of reasons. One being that, for example, if we are looking at live cell imaging versus some kind of endpoint where those are actually independent experiments going to different time points, then the model of the noise is necessarily going to have to be different, because we might expect more correlations between data points with live cell imaging than with endpoint experiments, for example. So really pay attention to, where is the data coming from, what kind of experiment is being performed here. Yeah.
One quick question. Are you doing this on the GR curves or on the survival curves? Because originally you mentioned you were going to look at GR curves, but like one of these outside the glow experiments, you have to take down the sample, so you're not going to have a time point 0. You just have the end point. For example, with the live cell imaging, are you then fitting to every time, or are you again fitting to time or?
Oh, I see what you're saying. Yeah. So for the selection of the parameters and the sort of inference that we're doing, we're fitting to every data point that we collect.
Okay.
So every time point, every replicate, every concentration. But in terms of analyzing the drug sensitivity of the populations that we infer, we're looking at the GR.
Okay. Now, given the number of parameters that you have down there, are they structurally and practically identifiable?
Practically speaking, I will show you our results right now. And we haven't done sort of a theoretical identifiability analysis, but I'm more interested in the practical identifiability anyway. So yeah, that's not something we haven't looked at a theoretical question yet. Okay, yeah, so to kind of get to how does this work? So the first thing that we did was we created some synthetic drug screen data, so simulated data to see how this thing works out. So what you're looking at on the left is the true underlying mixture percentages of mixture at the beginning of the experiment. We have 1, 2, and 3 populations, and then in the next column is what our method estimates. And then over here with these sort of lines and funny boxes are the true and estimated GR50, so that's drug sensitivity metric. And so what you can see is that, it's doing pretty well for 1 and 2 populations, and still fairly well for 3 populations, although we were seeing some wiggles in the GR50 down here. But the frequency and number of subpopulations is always correct, and the frequency looks pretty good. And I should say here that we simulated this data, and so we got to choose how much noise there was in the data, and this was standard deviation 5% of the initial cell population. And I'll talk in a minute about whether that's realistic.
Would it work with not with the subpopulations with a very distinct and narrow drug sensitivity, but something more realistic with sort of broader kind of range How well defined your subpopulations? Because oftentimes, like you look at the cells, for example, direct naive cells, it don't necessarily have the kind of genetic heterogeneity, but there is a distribution in the sort of growth rate for colony sizes or sensitivities.
Yeah. So, great question. We did look at a lot of questions, and I'll kind of get to that in a bit, like how distinct do subpopulations have to be in order for our method to pick things out, and also how small can the frequency be? Like if you have one cell in your sample that's different, we're not going to pick that up. So as you might expect, there's a threshold for all of these questions, and so I'll get to that in a couple of minutes. So this slide kind of focuses on, what if we increase the noise in the synthetic data? So now we have 10% noise here and 20% noise here. And so you can see that 10% noise is still looking pretty good. Again, a couple of wiggles in the GR50. 20%, we're starting to see like larger wiggles. All along, though, the number of subpopulations and the frequency still looks pretty good. And then down here, and sorry, this is kind of small, I just wrote down some estimated noise levels for some common experimental techniques that we were looking at. So for ViCell, we saw some of these are published, like less than 5% across several lines. Celomics array scan was reported at 1 to 5.6%. And so it's actually some of the data that you're going to see is coming from the Celomics array scan. So we felt pretty good about, kind of using 5% as our standard model or baseline model. Okay, so next we took this to cell line experiments. So again, you're still looking at true mixture, then estimated mixture, and then the comparison between the, I should say true in sort of quotes, so sort of intended mixture, right? So, but we didn't do kind of like any kind of testing after the cells were mixed to really quantify how many of each cell type was this. So this was sort of, the intended mixture. And so on the left, you're looking at some murine cell lines, isogenic, BCR-able with and without P315I, and they were tested in response to imatinib. And this was done by time-lapse microscopy on the Inky site. And then on the right, this is some known mixtures of non-small cell lung cancer lines with differential response to erlotinib, and with the resistant cell type having the T790M mutation. And so, as you can see, the method performed pretty well. In most cases, pinning down the number of cell populations correctly each time, and the mixture percentage almost every time, and the GR50 looks pretty good. You might be wondering why the bar here is so wide, and that's simply because we only had data at these 4 concentrations for these experiments. So of course we're not able to know where the true GR50 is within that bar. Okay, so then we're feeling pretty confident about going to clinical samples we looked at.
Can I ask another question?
Yeah, sure.
If you don't know how many subpopulations you have, can you reverse engineer that?
Yeah, we did not tell the method in any of these cases how many subpopulations there were.
Okay, so just based on the response data, it reverse engineer itself if there was only one population in there or 2 or 3.
Yeah, I don't know if maybe I skipped over it too quickly, but the sort of step 3 to the statistical framework, the model selection, is where we pick out how many subpopulations there are.
Okay, so you run a model for 1, you run a model for 2, you run a model for 3, and then you see which one is the most likely.
Exactly.
That's cool. That's very cool.
Does it matter if there's plasticity here?
Yeah, it matters. So let's see. Let me go back. I'm not going to get to that because I've got another story I want to talk about in a second out of the talk.
I won't derail this.
I'll just.
Clearly 3 could be
Yes.
not 3, right?
Yes,
One.
yeah, and we're actually, we're, so we have, so I'll just mention really briefly, like here, the interaction term that I'm not gonna talk about a lot in this talk, can, you know, this term here is sort of representing like, okay, what if a J gives rise to an I? And then there's also, you know, what if the presence of J's impacts the growth rate of I's and things like that. So all sorts of interesting questions we've started to kind of build out here. Plasticity is one. Macrophages being one of the cell types is another one. Lots of fun things to think about, and we're sort of I think we have a lot of the method kind of worked out, and we're sort of looking for good experimental platforms to test some of these interaction models. Okay, so on to clinical samples. So we looked at multiple myeloma, and I am not going to go through the sort of process of how these cells are sort of isolated and prepped here, because I will probably get something wrong, but these are the people who were involved in it from the University of Oslo, and they will know a lot more than me. But I just want to get to the outcome of our deconvolution analysis here. And so I'll just say, of course, we don't know the underlying truth here, so this is just what the method is telling us. So you're looking at drug screens from 5 patients treated with different drugs, and they were not all, the samples were not all treated with the same set of drugs, which is why you see each panel is a different drug, and then the different rows are different patients analyses. So what we find is that for all of these drugs, the analysis indicated that there are either 1 or 2 subpopulations for each patient. With the exception of dexamethasone, we see that there's sort of similar subpopulation GR50s across patients, but sometimes with varying mixture frequencies. So for example, here, for venetoclax, you can see, you know, all 3 patients kind of have a more sensitive and a more resistant component, but at maybe different frequencies. And in some cases, I think for this patient, this green patient here, we were able to obtain some genomic analyses of some of the known resistance mutations here, and it turned out that the mixture frequencies were consistent with the frequency that we estimated as well. So just for kind of like a proof of concept thing, we decided to sort of ask the question, okay, so if you have, you know, if you start treating these patients, and you know that you're going to treat with venetoclax, and that these patients have, you know, different heterogeneities, essentially. Maybe they have the similar behaving clones, but they're starting at different frequencies. Then how does that impact their disease progression if they're on the same treatment? So we used
I'm not fully understanding that slide before.
Sure.
Because I was tweeting and not painting. But just to clarify, you've got the samples from these patients. Were they treated with all of those drugs, or is it samples you then treat with drugs, or? Oh,
there's samples that we then treat with, not we, but my collaborators treat with drugs. But they're not all the same. They're not not all samples were treated with the same set of drugs.
That's why
kind of
there's
confusing
different
thing.
sizes on the
Yeah.
numbers.
Exactly.
And the different color is a different drug? Or
Different,
patient?
so like each panel is a different drug,
Right.
and then each row is a different, so each color is a different patient. You can see the.
Oh, I see it down on the x-axis. It tells you the drug now. I just never saw it before. Thanks.
Yeah, sorry, it's too small to see, so that's my fault. Okay, so basically, I'm not getting into the mathematical model we use to model the myeloma dynamics, because that's kind of not the point of the story here, but just sort of the idea being that, if we look at a marker of tumor burden, M protein over time, and we, in the model, put all of these 3 patients under the same therapy, because of the fact that they're starting out with different heterogeneity or different frequencies of these populations of cells, they end up with very different clinical course. So just sort of, the statement on the clinical impacts of heterogeneity, even if you have the same sort of subpopulations if you start out with different frequencies. Okay, so then we kind of got into a little bit maybe more technical questions of experimental design. When we're doing these drug screens, if we want to do this deconvolution analysis to get more out of our results, what's better? Should we have more replicates, more time points, more concentrations? So what you're looking at here is, this is simulated data, error in the mixture fraction, and in the so we did two population analyses for this slide. B is simulated error in the GR50, and C is error in the GR50 of the resistant, so sensitive resistant populations. And so in the light blue line is if you increase, for example, the number of replicates, how does the error decrease? And then the green is if you increase the number of time points, how does the error decrease? And then blue is concentrations. So kind of just to summarize all these plots, what we see is that, increasing the number of concentrations is probably the best thing to do. Increasing the number of replicates is not going to be that informative for this deconvolution analysis. And time points is maybe someplace in between. Yeah, question?
With concentrations, is that expanding the range or in the same range sampling more?
That's a very good question. We did both. I think this plot is in the same range, sampling more. Expanding the range is, I should say, very important, though. If you are, say, not sampling in the range where you see that dip in the Hill function, that's going to be a problem. So that's something to take into consideration. So this is sort of assuming that we're covering the right range, but how much more resolution do you need?
Now, Jasmine, going from 5 concentrations to 17 is probably a dollar amount attached to that.
Right. Yes.
And what is on the Y? I can't read the Y-axis. So what's the cost benefit for that? How much reduction in error do you get by increasing your concentrations? And is it both?
Yeah. So the Y-axis is like absolute error in the mixture fraction, and then this is like a log ratio. So it's a little bit more. Probably like take a little bit more time to explain like what exactly the errors are. So I don't know that I can give you a great answer on cost benefit analysis right now, but I would also say that the benefit kind of bottoms out like at 9 concentrations. So it's not going to be that beneficial.
Yeah. How generalizable are these things? So like for some drugs, for example, time points. At time points, there's like very high correlation between different time points. There are other drugs where there's like no difference at 3 or 4 days and huge difference at say like 2 weeks after the exposure.
Yeah, I guess like time points, I would kind of have the same kind of general philosophy as concentrations. Like if you have the window correct where you're seeing most of the action, then like this plot is really about like how much more subsampling you should do within that window. But if you are, you know, sampling days 0 to 3 and all the stuff is really happening, all the killing is happening on like day 10, then that's going to be a problem.
So it requires sort of a priori information about what to expect. Yeah.
Exactly. You kind of want to know like generally where you want to be. So basically, a couple of things I'm not showing here. We looked at small mixture fractions. So if you have a two population mixture, how low can you go for the small population, and 3 population mixtures. So we looked at some of those detection thresholds. We also looked at how close the GR50s can be for 2 subpopulations for us to pick it out. Unfortunately, I didn't put that one on this slide. But as I mentioned earlier, there is a detection threshold for every one of these questions.
This is super cool analysis. Sorry to interrupt. If you mix a couple of those, so if you go from 3 to 5 time points, 3 to 5 concentrations, do you get added benefit reduction error?
That's a good question. We didn't look so much at like the sort of higher order, like yes, we're going to mix some of these. So Evan, the student who produced these, I know did a ton of these kinds of plots, and you know, a very small fraction of them ended up being included in the paper. But I could definitely ask him if he what his thoughts are on mixing and matching. Okay. Oh, and then one last point I just wanted to make is that, you know, you don't see a ton of change as we move to the right for the resistant GR50, and that's going to be because you know, as you're applying the drug, your sensitive population is disappearing. So that's the one that's hard to capture. The resistant population is not so hard to capture. Okay, how much time do I have? Not a lot. Okay.
Not a lot now.
You get 3/4 of an hour you want.
Okay.
I mean, practice book until 1:30.
All right. Well, I guess Sandy and I are the only ones who are keeping from our lunch. So. Okay. So the second part, I'm going to be talking about joint work with Einar Gunnarsson and Kevin Leader, and in here we're going to be looking at switching gears and thinking about genomic analyses and what we can get out of that kind of data as opposed to the sort of phenotypic drug screening data. And so I don't think I need to introduce this slide to anyone here. We all know about, you know, different models of evolution in tumors and the importance of trying to figure out what exactly is happening, and also probably are all aware of ongoing debates about, you know, genomic tests of neutral evolution and things like that. So super interesting conversation going on right now in this field about these kinds of questions, and so we got interested as well. Right, so given the genomic data on the current state of the tumor, can we infer its evolutionary history? And so, for example, has it evolved neutrally, or is this thing under selection? And what are the parameters of the evolutionary process? And so the site frequency spectrum is a popular way to look at these questions. And if you're not familiar with it, that's fine, because I have a slide explaining it. But I'll just mention that this is not a new thing. It's been used for neutrality tests in the evolutionary literature for a long time, mostly in fixed size population models, so models like the Moran and the Wright-Fisher. Okay, so the site frequency spectrum, what is it? Let's say we start out with a tumor cell at the root of this tree, and it gives rise to 2 daughter cells. And one of the cells has a mutation, which we'll call A. And then let's say A gives rise to a progeny, and that cell picks up another mutation, B. And then so on and so forth. So these cells continue to pick up mutations, some of them die, so on and so forth. At the end, right? So let's say this is the time point that we get some data. The site frequency spectrum is going to tell us the number of mutations that are found at any given frequency in the tumor, right? So here, for this particular tree, we have 4 mutations that appear in just one cell, right? So those are the mutations E, G, H, and I. There are 2 mutations that appear in exactly 2 cells, and so on and so forth, right? There are some mutations that are found in every single cell. And so here, A and B, mutations A and B are both found in every single cell. And those would be called clonal mutations. And of course, a lot of the time that happens if the initial cell has all of those, has those mutations. But as you can see here, in this case, that isn't the case. A and B came up after the initiating cell, but everything else died. So that can also contribute to clonal mutations. Right, so one thing that should be sort of pretty obvious when you look at the definition of site frequency spectrum is that you're losing a lot of information, right? Like if you actually have all this data, but you're only going to analyze this histogram, you know, you're losing a lot of structure here from your analysis that, you know, could tell you something about the evolutionary history of the tumor, right? So that's my big caveat to this part of the talk, is that the site frequency spectrum may or may not be the best way to get at these questions. Nevertheless, it's widely used, and we decided to think more about how can we get the most out of site frequency spectra.
If you have this histogram at every generation, can you uniquely reconstruct the tree?
That's a good question. I don't know that, but that's a good question. To think about that. Of course, we don't have it at every generation, and also the generations are all overlapping, so.
And mutations don't always occur during the generation.
Exactly, yeah. So. Okay, so kind of before I get into some prior results, I'll just give you some notation and a model that we're going to use to kind of start thinking about the site frequency spectrum. So let's say we have the Z naught being the size of the tumor population at time t, and we start out at 1. And we have a standard kind of birth-death process where each cell divides at a rate R naught and dies at rate D naught. And then we're going to be able to accumulate neutral mutations during cell division in this model. And so it's going to be, you know, you accumulate some random number of neutral mutations. We're going to use infinite sites. And the average number, so I'm not specifying what's the distribution of this random number of neutral mutations. All I care about is it's got an average of W, neutral mutations added per cell division. And just a little bit of notation here. Omega infinity is the event that this thing does not go extinct. And P naught is the death rate divided by the birth rate, and that'll come up a lot in the talk. And Q naught is just 1 minus P naught. So P naught is the extinction probability, 1 minus P naught is the survival probability. I will repeat that so that you don't have to keep that in your mind. Or feel free to stop me too if you forget. Okay, so what's been done before? So a couple of different papers have derived some approximations to the site frequency spectrum already. So one of the sort of more well-known formulations is if we let M sub S be the number of mutations found at frequency bigger than or equal to F at time t, then as t goes to infinity, the expected value of this quantity looks like this. So it's W's, remember W is the average number of neutral mutations you accumulate during a cell division, divided by Q naught, which is the survival probability, and then times 1 over S, so S the frequency, minus 1. And so one of the things to to note about, so I should say that this was, I believe, first derived by Rick Durrett in 2013, and then subsequently re-derived in a couple of other papers, and I'm probably missing some here, so I apologize. So one of the things to note about this result, the 1 over F law, is that it's an approximation that was obtained using what's called the skeleton process. So the skeleton process is if you take this tree, so if you run this for birth and death process, you get a tree like this, some things die, some things give birth, and you only consider the lineages that survive forever. That's the skeleton process. So that skeleton process was used to make this approximation. And it turns out that that is only valid at large frequency, so for the large frequency end of the site frequency spectrum. So I'll come back to that in a minute. And then I'll just mention that you might have heard of the 1 over J squared law, which is essentially this is a cumulative version of this result. So Sj is the number of mutations found in exactly J cells at time t. So instead of doing at a frequency larger than or equal to F, now we're talking about a specific number of cells. And so there, if we look at the effective value of that quantity, then we get something that's 1 over J squared. So one thing that's important to note is that these two parameters, W and Q naught, in both of these formulations of this 1 over J squared law, are stuck to each other, right? We only have the ratio W over Q naught, right? So if we want to know what is W and what is Q naught, we are out of luck with this 1 over J squared law. Okay, so what have people done? So Williams et al. In 2016 suggested a neutrality test based on a goodness of fit between large frequency mutations and this 1 over J squared law. I'll talk more about that work in just a minute. In that paper and some other papers, various groups used this 1 over J squared law to estimate the effective mutation rate. So they're calling that this W over Q naught. So again, these two parameters kind of stuck to each other. And as I mentioned, we cannot separately identify the mutation rate and the survival probability using this law. Okay, so this is plots from the Williams et al. Paper. So just to kind of go over the gist of what they did. So this is like the site frequency spectrum, the histogram, and on the x-axis is the allelic frequency F, and then on the y-axis is the number of mutations. So what they did was they said that any mutation that is present in more than half of the cells, so that's going to be allelic frequency more than 0.25. They're going to call clonal, so that's going to be out of the analysis. And then any mutation that's present in lower than 0.12 frequency is going to be out too, because they don't believe that because of the detection threshold. Okay, so they're looking at the part of this that's between 0.12 and 0.25, and what they...
Noemi?
Sorry.
So they're going to look at this section of this, so sort of what I would call the large frequency end of the site frequency spectrum, and test the goodness of fit with this 1 over J squared law. And then they did this in a number of different tumor sets, the sets of tumor samples, and then used that goodness of fit test to sort of say that 4 out of these 7 tumors were evolving neutrally, and 1 out of these 82 tumors were evolving neutrally, and so on and so forth. Okay, so that just sort of gives you an idea how high frequency spectrum is used to kind of look into the tumor evolution questions. And I should mention that there have been other works kind of after this that kind of added to the conversation, maybe, and I probably can't summarize all of them, but that, you know, for example, I think, you know, some people have sort of pointed out that, you know, if you have a tumor evolving with selection, then if you calculate the probability that you will be able to see that within the sort of narrow 0.12 to 0.25 range, that's quite small. So I think that was work by Ivana Bozic in I don't know I'm gonna do this on that. But anyway, but there have been lots of interesting papers kind of spurred by this seminal paper. Okay, so the prior work on the SFS is mainly based on approximations. Basically, the skeleton process approximation is focused on that large frequency end of the spectrum. And our goal is to gain a more complete understanding of the SFS at both small and large frequencies. And sort of as a byproduct, we have sort of a suggestion, sort of in principle, for a way of estimating W and P naught separately using the small frequency end. Okay, so I don't want to take up too much time. Maybe I'll skip some of the, let's see. Let's just skip to the results, because I started out 12, right?
Yes.
Okay, let me just skip a few things here. Okay, so I think if I just skip all the way to the results, one of the interesting things that we found was that in parameter regimes that are relevant to cancer, so this is going to be a high P naught value. So P naught is the death rate over the birth rate. This has been estimated in several papers. You see they're almost all above 0.9. So high P naught is what we think is a relevant regime biologically. What happens in those tumors, kind of morally, is that you have a lot of cell death, right? So the high P naught means high cell death. And when you have a lot of cell death, so let me actually take a step back. If you think about in a tumor, the mutations that are going to end up at large frequencies, it's typically going to be the ones that appear pretty early on, so that they can kind of surf their way up to high frequencies. And those tumors, those mutations happen on the skeleton for the most part. If you look at the mutations that are in the low frequency end, those mutations, a lot of them are they might have come up quite recently, right? So they may actually die out if we didn't take it out and sequence it. And so when death is high, that small frequency end sort of becomes more important, and your process starts to look more like a sort of critical branching process, or more like a fixed size population model. So what we found was that if you look at the high frequency spectrum for a high P naught tumor, then what you'll have is you'll start out at the large frequency end with the 1/J² law, but as you move towards the low frequency end, the spectrum will bend away from the 1/J² law, and then, so the 1/J² law is the red line, and the green line here is the 1/J law, which is actually the high frequency spectrum for a Moran process, a Ray Fisher. So we have this sort of bending down to a 1/J law, or deviation from this 1/J² law, at the low frequency end. And so we thought that was super interesting and perhaps hadn't been noticed before. And so Einar, our student, decided to come up with a method that kind of exploits that deviation at the low frequency end. And so this, at this moment, this is theoretical, and you'll see in a moment why. But in principle, it sort of gives a roadmap for being able to identify the mutation rate and the survival probability separately. And so the idea is that if you take the number of mutations in one cell and divide that by the total number of mutations, then if we look at our analysis, which I skipped over, so this doesn't make any sense, but if you look at the analysis, what happens is that that ratio is independent of W, but depends on P naught. So we can invert it, and so we can take the ratio of the number of mutations that are only present in one cell and the number of mutations, the total number of mutations. We can take that number and calculate P naught by taking the inverse of this function. And then of course use the rest of the site frequency spectrum to calculate W on this. Right, so the reason this is theoretical is because can we actually get the number of mutations that are present in just one cell in a tumor? No, we cannot. But sort of gives us a roadmap and some ideas to start trying, like, okay, what if it's not exactly one cell? What if it's a small number of cells? Things like that. So we're actually trying to think about now, you know, how can we take this idea and turn it into something usable with the current detection limitations that we have. Okay, and I think I will just, this histogram just kind of show that with simulated data, the method works, and you can identify the underlying P naught by using that ratio. And as I said, the limitations. But I will just sort of summarize by saying we derived exact expressions for the site frequency spectrum for a neutrally evolving tumor. And if you look at the small frequency end of the spectrum, there's a lot of interesting information there that you can, in principle, exploit to estimate the parameters of evolution. And with that, I will put up my acknowledgement and funding and say thank you.
A quick question about these estimations of the patterns of evolution. So does it have to rely on the assumption that basically everything that matters happens at the genetic scale? Because we know there are important, like, stable changes in gene expression that don't necessarily, they would not necessarily be reflected in specific detectable mutations, but that could have a huge impact on fitness. There's definitely microenvironmental effects where, depending on what part of tumor the cells land up, they're the same genotype can have very different consequences in terms of fitness, etc. Like, are these inferences useful independent on whether the action is happening on genetic level or non-genetic level, or is it based on the assumption that everything has to be happening under this lamppost of genetic changes?
Well, yeah, good question. So I guess we came, you know, our perspective on this project was, you know, there's a lot of people who sequence tumors and output things like the site frequency spectrum. So what can we do with that? What is the best thing that we can do with site frequency spectrum? That being said, you know, in our group, we're, you know, let's say majority of work are more focused on the kinds of things that you just mentioned, and we're not so much historically a genomics kind of group. So we did actually start to ask some questions like, okay, what if we have plasticity in the population, and how is that going to impact the site frequency spectrum? Are there signatures of plasticity in the site frequency spectrum? Things like that. So preliminarily, the answer to that question is no, we cannot. The site frequency spectrum, if you have some sort of like this sort of phenomenological model of plasticity where you're switching between cell types, there's some small changes in the site frequency spectrum, but it's not so much that you could really detect anything. But yeah, I guess in terms of non-genetic or environmental changes and being able to infer evolution that's sort of more driven by those kinds of things, we are working on things like that, probably more so than this line of work. But I wouldn't advocate using the site frequency spectrum for looking at those questions. Yeah.
This is probably very wrong, but I have to ask this anyway. You kind of were talking about looking at it from a cell perspective. So the idea that the number of mutations in the cell versus the number of total mutations can kind of help you equal P naught. What if we're looking at it from a gene specific, where we now have a population of people who have either mutation or not mutation in this gene? Could we then look to see and calculate the P naught and maybe infer whether that gene is actually under negative selection if it fits with? Is there a way to flip that onto a population gene level, the other way around?
Yeah. So I guess the general question is yes, and I think that people have looked at questions like that, maybe in the more sort of organismal evolution level. And with similar kinds of techniques, although I can't think of like any specific examples at the moment, but I would say yes, and that this is an area of research that I think has been investigated. Yeah. Sorry, I don't know much more.
It's very cool.
Yeah.
So, first of all, super nice work. I have a question about the P naught.
Mm-hmm.
If I understood your work correctly, then P naught is the ratio between the cell death and cell division, right?
Yes.
So, here's a question. If you have cells that have a net growth that is very, very fast, you have a lot of cells that are dying, so a lot of cells that are dividing, and you have a very fast-growing tumor versus a tumor that grows slow but has the same ratio. Are there any limits of how low the net growth could be versus the P naught? So I'm talking about the differences versus the ratio of death and proliferation.
So when you say limits, what do you mean?
Limits. So for instance, if we are looking at the accumulations of mutations over time, but if we have a tumor that is growing very, very slow, accumulates a lot of mutations throughout 70 years of a person's lifetime, would that be a limitation to your result? Even though the P naught might be having a nice value that fits.
I see. Yeah. No, that's not like we don't have the model will still be valid as long as overall the tumor is not shrinking.
Okay.
Yeah.
Cool. Thank you.
Yeah.
Yeah,
was very interested when your curve flipped from one power law to the other. It's super cool. And you emphasized a couple of times that your P naught is quite high for this analysis, and you give a justification that for some cancers this might be true. So how dependent is this transition between the power laws on the range of P naught? Do you get that for lower P naughts as well, or?
Do you have what?
Do you get similar behavior for lower P naughts as well, or do you need a P naught close to 1 for that?
Okay, the best way to say this is, if P naught is 0, then there's no death, and so everything is a skeleton process cell. So 1/J² law is followed all the way, right? So that's P naught equals 0. As soon as P naught is not 0, you will still have that in the large frequency end, you will follow 1/J². And we can see that from the analysis, the theoretical analysis that you will still have 1/J². But you will start to see deviations in the low frequency end. And then as P naught approaches 1, that deviation now approaches 1/J, right? So it's kind of a continuous bending.
So if you
the
have,
low frequency end only.
so if you have a much lower P naught, you just have to increase your number of cells to drag it along.
Well, if you have a lower P naught, it might not actually look like 1/J at that point. It might be like,
Right, you need to go much further out the cell numbers to see the profile, right?
I think you still wouldn't necessarily be hitting 1/J, even if your cell number was super high. So it's really like, I guess if we show the sort of like the analysis here, it's really a P naught goes to 1 result. This bending towards that's when you see it matching up with 1/J. But you will deviate before P naught goes to 1. And then we didn't do any analysis on how fast you see it go. How big P naught has to be for it to look within epsilon of 1/J. We didn't do that analysis. Yeah, that's a good question.
I have a couple of questions. So that was very interesting. I'm very interested in this work in general, and we tend to simulate more than do analytical work. But obviously, both are kind of nice and can talk to each other. And I like how you used synthetic data to sort of tune your approach and sort of understand it. I've worked a bit with Trevor, and he works in this space, and so that kind of his 1/F is kind of equivalent to this.
Yeah.
One of the things that became clear in the work he was doing was that rare mutations are hard to capture because sequencing isn't deep enough, right? And so your point about being able to sequence single cells will take in there. But in subsequent work, he was looking at selection and how the shape of that spectrum had developed bumps, basically, right? And particularly in that region that you were talking about. I assume that your approach can also capture that.
This is all a neutral evolution model. So, I guess in principle, that, going back and...
Going back to the start.
Yeah.
I understand that this is a deviating from what you're interested in, but if you can, there's that potential could be very intriguing because, understanding then being able to capture evidence of selection as opposed to that it's a neutral process.
Right.
So, the flip of that coin, so to speak. But a kind of more practical question comes back to the assumptions, and you kind of touched on it when we talked about plasticity, and we can discuss the plasticity question over lunch, but I'm more interested in the interaction question, right? right? So obviously the clones could be interacting with one another, which would shape their frequency, right? Does this model account for that?
This model?
Yes.
No. There's no I mean, this is a
Well mixed, sort of.
standard branching process model, yeah.
But is that something that you think could be worth looking at, or is it too complicated?
Worth looking at, yes, obviously. Very worth looking at. Whether or not it's too complicated to be mathematically tractable, not sure.
What kind of data would you need?
What's that?
What kind of data would you need to determine the probability of interactions between clones? It's hard enough with...
It feels like you've already got that data, it's just you've not looked at it through that lens, right?
Right, that maybe I mean, perhaps some sort of that would be an interesting, same way we were asking this question, is there a signature of plasticity or phenotypic switching in the SFS? We could ask the same question, is there a signature of interaction in the SFS? That would be definitely interesting to look at. Probably challenging, but definitely would be really cool to look at.
Because I think, from our point of view, that if we can get a handle on that, if we could pull that out, that type of data, because obviously the thing is, this type of data is abundant, because biologists measure that sort of stuff. Right?
Yeah.
So if we could pull out something like that, because from our point of view, one of the hard things to tackle or to quantify is interaction.
Right. Yeah, that's actually, I mean, so I definitely would like to talk about that more, and I mean, I think it's definitely worthwhile to see whether we can adapt this analysis to that question. I was just going to mention that, we are in sort of in the first part of the project, the deconvolution, also asking that exact question right now with the drug screen data. Can we detect interaction between subpopulations from that data, which
that might be more easy.
Might be easier because you've got dynamic.
The temporal dimension there really helps, right? So we do that a little bit where we assume there's a sensitive resistance. But if you've got, say, there's 3 clones or there's 4, or they're switching between each other, right? Then it all gets a little bit messy. But I was just thinking more explicitly of this analysis.
Yeah. I mean, there could be some intermediates too, right? Maybe you can't just from one single snapshot SFS tell okay, well there's 3 clones and this one is doing this thing to the other. But maybe you can say yes, there is some interaction of this class in those populations.
And you might get, say, 3 time points.
Yeah, that would change things a lot. I mean, I think even for this particular analysis could be changed.
Often you'll get a pre and post.
Okay.
Treatment type of sample or something like that.
Okay.
So that would give you a kind of what it was like before and what it was like after.
Yeah, that could help a lot. So yeah, I'd like to hear more about it.
Cool. Well, thanks once again, Jasmine.